Hi everybody. All right, so we're going to start with our last video for exam 5. Um, before I start, let me go ahead and say this test isn't going to be your hardest test at all. It should be fairly straightforward. I'm not going to go so far as to say easy because there are a few concepts on here that could get a little bit confusing. But what I will say is that it's going to be similar to some of your other ones in that you're going to have a lot of case scenarios for your short answer, because he does talk about ethics in his last slide, and I could see him bringing that up in a lot of multiple choice questions, and maybe about one short answer question, not too much after that. So, for multiple choice, it would be, which one of these resembles an ethics question, which, okay, which, which principle does this scenario follow, which solution would be best, that type of a thing. Would be multiple choice or short answer, and I wouldn't expect too much of it. So I know some of you guys were really excited that ethics was on there, some of you weren't, but just to give you guys a heads up, I wouldn't expect that to be a large majority of the test. The other lectures about quantitative trait loci and things like that will play a bigger part than the other stuff will. All right, so let's go ahead and get started and go through this packet. All right. So it starts with lecture 19. You guys had talked about this for the last exam, what forward genetics and reverse genetics were. I don't know how much in depth. I don't think you guys got that much in depth for last test, but this test, he kind of briefly goes over it again. Just know the differences between those two. That's going to be important. And what they are hindered by. So if you go down to, it's bullet point number 3 under genomics A, it says both of these processes involve an ample knowledge of genes, which is why the process of forward and reverse genetics were slow going until recently. And then where that arrow is, I would highlight where it says both methods are severely hindered by the requirement of knowledge about genes and phenotypes, because that's what makes these processes take a long time, because sometimes these libraries that we've created aren't enough for us to figure out what genes do what. And not only that, but if there are surrounding genes that we don't know their function, it kind of puts us even more in a hole, because we're trying to find the function for one gene, but we need to use genes that we still don't know the function for. So it makes it kind of complicated. Going down to the brief history, I put the definition of contig there because some people who I've been tutoring have been really confused by that word, and it's so understandable. If it wasn't talked about as much, it can get confusing to hear that over and over again. So contig is just a shortened form of contiguous, or it comes from the term contiguous. So it's just a set of overlapping DNA fragments that together make a consensus region of DNA. And so that's what we're talking about when we're talking about contigs. I threw that in there just to give you guys a little bit of background. Human Genome Project, I would know not everything about this. I don't think you need to know the dates per se, but you should know that it took so much longer than everyone was expecting. Everyone was thinking, all right, we can do this. We're all ready. We can do this. We can sequence the whole human genome. It's not going to take too long. They projected that by 1998, they'd have over 50% figured out. But if you go down and look at that number 1, it says 1998, halfway through the project, only 4% of the human genome was sequenced, which was just so low for compared to what they expected. So then under that, so you should know what the idea of the Human Genome Project was. So they wanted to sequence the genome of an entire human. It's pretty intuitive. So know that definition. And then I would also know clone-by-clone sequencing and whole genome shotgun sequencing, which we'll get into in a second. And yes, you do need to know those processes. Not so much in depth, you don't need to be able to, draw out a mechanism, but you should know the basic steps, which I've underlined here. I didn't go super in depth. I didn't, put how they do it or anything like that. It's just construct physical map, clone genome into a lot of pieces, subclone, and then after it tells you all the steps, there's a figure in between clone by clone on the next page, so on page 3, that shows you clone by clone on one side and whole genome shotgun, so you can kind of visualize the difference if you're more visual like I am. So So yeah, so that's clone by clone. You guys can read through that. Moving down to whole genome shotgun sequencing. So what you should also know is that clone by clone sequencing is technically difficult. So I would highlight where it says technically difficult in the 1998 bullet. And then for whole genome shotgun sequencing, it is computationally difficult. So just know those two differences. And the main obstacle for whole genome shotgun sequencing is repetitive DNA. And it kind of goes into that. You can read why that is hindered by repetitive DNA there. All right, moving on to page 4. I want to keep this video kind of short because I know you guys have other finals to study for. I have other finals to study for. But I'm going to give you guys basically everything you need to know for the exam. I'm not going to read. Well, I don't usually, but... All right, so how can we put the numerous contigs together? Know paired end sequencing, know the not the mechanism, but know how it's done. So the purpose, how it helps when it can't be used. So like under bullet 2, it says this approach can't be used in cases when repetitive elements are longer than the largest available clones. So just know these bullet points, know what large insert paired end libraries are, what a supercontig is. Going on to the next page, it tells you under the second bullet from the top. And then I've just included a diagram for you guys to see how paired end sequencing kind of the scaffolds are assembled. And then when we go down to here, in summer 2000, the genome was finished. So, but there were several gaps. So I would know maybe summer of 2000 when the genome was considered pretty much finished, and then it was completed a couple years later as they were filling in the gaps. Okay, so let's move on to the next page, page 6. Okay, yes, so these two examples, he gave you an example of whole shotgun sequencing or whole genome shotgun sequencing of a bacterial chromosome. I would go through and know the information so that, Haemophilus influenzae was the first genome to be sequenced. That's a fair true-false question. He could change the name to like Haemophilus Harry Potter, I don't know, and you would need to know that like that wasn't the first genome. So I would just memorize that. That's pretty easy. 2 types of gaps. So you need to know the difference between a sequence gap and a physical gap. You need to know which one's harder to fix. Physical gaps are much harder to fix than sequence gaps. Sequence gaps can be created from other strands of DNA and used to fill in missing pieces. Physical gaps are much harder because they're very distinct. So they're unique portions of DNA that don't have clones that we can make to fill those gaps in. So it's very harder. It's very hard, not very harder. And then B shows you how to close the physical gap, so just know how that's done. Yeah. So moving on to the next page, this again is just a diagram illustrating, what was going on during that whole process of the Haemophilus influenzae genome. I would read through that. You don't need to know exactly what happened, but you need to know that physical gaps, or gaps in general, sequence and physical, occurred after the production of that scaffold, and kind of how those are fixed. So how you resolve that problem, because that could very well be a short answer. If you have a physical gap, or if he shows you something and you can tell that there's a physical gap versus a sequence gap, he may ask, okay, how do you fix this problem? Multiple choice for sure. Possible short answer, I would say. Okay, whole genome sequencing or shotgun sequencing of a eukaryotic genome. So I would know that Drosophila melanogaster was the first large one. I wouldn't recommend knowing that over Haemophilus influenzae being the first one altogether. But you can kind of keep this one in the back of your head for a possible true-false. So yeah, you guys can read through that. Again, they had, here they had large scaffolds and small scaffolds, and it kind of goes into what the differences were between those. So de novo genome sequencing, I'm on to the next page. I'm on page 8. Sequencing is now super cheap thanks to massively parallel next generation sequencing. Regular genome sequencing is just a brute force approach, but the newer genome sequencings that are coming out are just so much faster. They're able to read the libraries and pair them and kind of circumvent having to figure out genes and things like that. So, yeah, it's just easier. Okay, moving on to annotation. This you're just going to have to read through. You do need to know what annotation is. So you're just attaching functions to DNA sequences and then trying to figure out what they do, what genes do. It's similar to, forward and reverse genetics, except annotation is when you identify the location of the genes versus actually what they're doing. So you're identifying the location, then the function, and then it helps to figure out not just like the physical function, so not just what phenotype you're getting, but it identifies the biochemical, chemical, and all that kind of stuff. So like the real like background of what we're trying to figure out. That's what annotation is. So the approach, it kind of underlines it underlines the experimental approach. I would know that. So D bullet point 3, and the computational approach on page 9. I would also know that. Let's see what else. Non-protein coding genes. Those are very difficult to figure out using annotation. Computationally. You can use it experimentally. It's a lot easier. So that's a good example to know that non-protein coding genes are the ones that can't use both approaches to be figured out. All right, moving on to gene families and how they arise. So you need to know that they arise via gene duplication. It says that under the second bullet point. Moving on to page 10. You need to know what the fate of a new duplicate could be. Know the definitions for non-functionalization, neo-functionalization, and sub-functionalization, and what each of those entails. You also need to know the next bullet point, why aren't all duplicates silenced? That's a great short answer question. He could just throw that exact question on there and expect you to be able to write an answer. And it could all be a part of a larger short answer, but again, it's fair game for the test. Frequency of gene duplication, I would know this at length. It's not that you need to know exact numbers. Births and deaths of genes, this is very good to know how new genes come about and how they die off. So again, for multiple choice reasons, I would know that Drosophila became a major source of new genes, for gene duplication, that during testing, 80% were new genes, 80% were like old, like normal genes, and then 12 were new. So moving on to the next part, page 11. Variation in genome sequence among species. Okay, so this is where we're going to kind of get into, or this relates to the human-chimp stuff. But first, we'll look at gene number and gene density. So genome content, under bullet point A, I would highlight genome content predicts cell size, and cell size predicts body size, because that's going to come into play down here where we talk about the C-value paradox. So the idea was that the amount of DNA in the haploid cell of an organism isn't related to its evolutionary complexity. But what they have found as an update to the C-value paradox, since those two aren't related, they found something that is related to determine complexity of an organism, and that's the gene number paradox. So this says that the gene number correlates to the complexity of an organism. I would know both of those. I would know the C-value and the gene number paradox, and also that sentence that we underlined or highlighted, whatever, up on this page as well, because it would be a good question for him to ask you from a short answer standpoint, what derived from the C-value paradox, what does it mean? Just a bunch of different questions can be asked just from these two things, and I think they're very important for your exam. I would say for maybe like 1 or 2 questions, but still valid for a short answer. And again, you'll see that as a question in the short answers that we're going to send you. And again, expect those at noon today. So yeah. All right, moving on to lecture 20. Genomes are fluid. So there are numerous inversions, translocations, fissions, and fusions, etc., even reasonably closely related. So the whole point of, I think, him including this in, some slides is just to show you that there's so much variation that can occur in a genome, which I know you guys know because you've seen meiosis, mitosis, all those things, and all the processes of the body and how mutations arise so frequently and things like that. So you know the genomes are adaptable, they're fluid, they are... no one genome is ever exactly the same sometimes, and things like that. Also, something that's interesting, so synteny, you should know what that is. Moving down here to centromeres move. This is going back to looking at the different types of centromere arrangements, so metacentric, submetacentric, that type of a thing, telocentric, and I think acrocentric. That one isn't pictured here. Just kind of review those and know that centromeres do move along a chromosome. Okay. So now moving on to the human-chimp genome comparison. I'm sorry, the phone is ringing, but I can't reach it. Okay, so we're gonna keep going. I got a lot of questions from you guys about how this is gonna be asked on the exam. Again, it's really interesting stuff. I just don't think you need to know the super hardcore details of it at all. You should just know, I think that the most important things to know from this are that humans have one few. Okay, so I'm on page 12, under bullet point, or under A, I'm on bullet point number 2. So humans have one fewer pair of chromosomes than apes do. So humans have 23 pairs, apes have 24. And the reason we think this happened is because two ancestral ape chromosomes fused together to form human chromosome number 2. Know that. I mean, that's pretty simple. It's pretty straightforward, but I think that's basic information that you should know. And you should also know about the FOXP2, the language gene. It's present in us, but not in apes, and it's required for the proper development of speech and language. And they think that comes from the fusion of those two chromosomes that apes have to create our chromosome, our one chromosome, which is human chromosome 2. So I would know those things. I included some diagrams that he had in his slides just because I do think that there's a possibility that in your short answer there will be a diagram that he provides you and he wants to know, or he'll ask you a question with it. I was telling somebody earlier that, he's never going to put a diagram on there and be like, hey, figure this out. It'll always be in regards to a question of something. So you just need to know that there are a lot of there's one huge chromosomal difference between us and apes, and we just talked about that. So now we get into how we've gotten our, how studies have been done. So how, we tested for divergence, but that gave little insight into relevant differences between two species. And then it just tells you some of the results that were gotten as well. This diagram is showing you just the sequence lengths. So ones that are present, but in the chimpanzee, but not in the human. So since this is printed in black and white, I guess it's kind of hard to tell which lines are blue and which ones are red, but you can always go back into your slides and kind of take a look at it. It's good to know or to, just see an example. You don't have to memorize these facts, but I would know that the prominent spike around 300 nucleotides corresponds to SINE insertion, so SINE insertion events. I would pick up on those things when you're reading these graphs so that you can understand what's going on, because if he puts a graph similar to this on the test and you see a spike and he asks you, what could have caused this, it would be good for you to have some background information to help you out. So you've seen this before, you could say. So you don't need to memorize that, but just need to try and understand some of these diagrams that are in there. Okay, moving on to page 14. Okay, so again, this is just another diagram that you can take a look at, looking at deletion size. Moving down to the 1,000 Genomes Project. So this was to establish a very detailed catalog of human genetic variation. So scientists had planned to sequence the genomes of at least 1,000 anonymous participants from a number of different ethnic groups within the following 3 years. That's about as much as I would know about that project. Moving on to quantitative multifactorial diseases, I would know a handful of the congenital and a handful of the adult onset and know the difference between the two. So congenital are obviously diseases that you get from the womb. So it's just you're born with them. Adult onset are ones that, you're born with inheritance of, but they don't appear in you until you're an adult. So adult onset, pretty intuitive, but just know the difference between those two and know a few examples. Lecture 21, Quantitative Trait Loci. Okay, this is by far probably one of the most important lectures to understand that we've talked about so far. So quantitative trait loci, you're going to want to know what they are. So they contribute to the variation. Know Fisher, how he was like, there are going to be a ton of these and they're going to have a very small effect. But it turns out that there are actually a few of them and they have a very large effect. So know that differentiation. Genome-wide association studies, you're going to want to know what those are, and the best definition, the most quickest, concise one, is under bullet point B, under genome-wide association, and it says it scans the entire genome for associations. That is like the basic definition that I would say you need to know. And I would just read through all of this about linkage disequilibrium. Everything on this page 15, is very important for this exam, everything on here. I'm like looking at it right now and I have writing all over this page because I want to say different things about everything, but I'm just going to go ahead and say, know this. Let's see. Quantitative trait loci mapping, know how that's done. So the mechanism is kind of written here. It's pretty short, it's pretty simple. That's one that I would know. I told you guys about some of the other ones in the earlier lecture that you just kind of need to know the basics of it. So like clone by clone and whole genome and that type of a thing. This one I would actually know the process of. Okay, moving on to page 16. That was a pretty short recap of that lecture. So lecture 22, we're looking at, how these qualities are after research, how the data is presented. That's mostly what we're talking about here. So, you've got, cross out under bullet point 2 that says 2 types, because we only talked about one. We only talked about, and we're only talking about continuous, so that makes sense, but just cross out 2 types. There are 2 types of curves that you can have, but you only talked about one, so that shouldn't be there. So quantitative metric traits, as opposed to quantitative multifactorial diseases. These traits are characters that can take numerous possible values. So your height, your weight, your IQ, whatever. Those are quantitative metric traits. I would just know that. Continuous traits often show characteristic distribution. Okay, talking about this bell-shaped curve. So this is just describing to you what the center of the bell curve is. A lot of you guys know this from like basic algebra, from whatever, but it's just kind of re-illustrated here for you. As well as mean mode variance. I kind of like went back over those definitions with you guys because from a mathematical standpoint, you probably remember it, but this is looking at it from a statistical standpoint. And so Ronald Fisher, who we had just talked about a second ago, he discovered how inheritance in continuous characters works together, and he basically invented statistics by accident, kind of, because Galton and Mendel, like, their theories were kind of going a little bit head to head. So Galton believed in the regression to the mean, but Mendelian inheritance, we've got those phenotypic classes and things like that. So it's not necessarily a regression to the mean. It's just you're having recombination events that are occurring. There's Mendelian inheritance occurring. So Galtonians, scientists who strongly sided with Galton, and then, or Galton, however you say it, versus Mendelians, people who strongly sided with Mendel, and it kind of illustrates their differences here. So the central limit theorem, I would know that. It's D2, central limit theorem. I would highlight that because that's something you need to know. Okay, moving on to page 17. I just put a little review of the Hardy-Weinberg equilibrium that he had in his slides on here because just to refresh your memory, in case you get something that you have to calculate on the test. Again, if you do, it'll be very straightforward, and the only thing you'll need to know is how to decipher the data from it. Again, it's not going to be too hard. It's going to be straightforward. We'll include a sample problem too of this. And then you also need to know, you just need to know the equations that you need to use. All right, quantitative trait genetics. It's a good experiment to know, the one that Edward East did. I think the most important things to know are the two questions that stemmed from it. So, why is there variation among pure breeding parental plants, which should be homozygous? What he's trying to get you to understand here is that the environment and non-genetic effects can play a big role in how, humans are developed. And sometimes that's a little bit overlooked because we're just thinking about genes, cells, how do these, work together? But it's really, the environment does play a big role. So moving down to quantitative trait variation. All right, so you need to know these basic equations that total variation is equal to phenotypic variation. Those are just the same values. And then phenotypic variation equals genetic variance and environmental variance. So we're going to actually have a problem about this. You guys can read through what genetic variance is and environmental variance, but yeah, we're gonna have a problem about this because I feel like it's gonna be easier to explain to you through you solving it and me like showing you the answer after you're done than it would be for me to say it right now anyway. But basically, genetic variance, amount of phenotypic variance due to genotype differences. If everybody has the same genotype, great, genotypic variance is 0. But environmental variance is the amount of phenotypic variance due to variability in the environment. And of course, environments are so variable, it's almost impossible to control an environment so well that you have organisms living in so that the value is 0. You're going to get around 0.3, 0.8. You can never get it exactly at 0. Okay. Moving on to page 18. This is showing you those values using the corolla length experiment. So like I told you, you don't need to know the experiment. I would know it in the context of being able to understand this information, but I wouldn't know it as in there's going to be a question about it on the test. So read through what I've put there for the corolla length experiment just so you know the background and like what was going on, and then use it as like a practice problem to understand what VP, VE, and VG are. Okay, so threshold traits, having the condition versus the unaffected, those type of a thing. I would just read through that. So threshold traits are traits that have distinct categories. Okay, and that's about that. There's not much to talk about with that because I don't think it's going to be that important for your test. Okay, heritability. We get into some pretty basic stuff here, but the two main keys that you're going to need to know are broad sense versus narrow sense, and you need to know those equations. So let's go ahead and move straight on to that, actually. We're going to talk about broad sense. So within a population, we could ask what percentage of the variation that we see is due to genetic variation, or what extent is due is the trait heritable within the population. And so that equation is big H is broad sense, equals VG over VP. So that's genetic variance over phenotypic. And then we've got narrow sense, which you need to know these 3 basic different types of variance, so additive, dominance, and epistatic. And then you just need to know the equation, which is little h squared equals additive variance over phenotypic variance. Okay. Oh, and yeah, so you would need to know that the interest is usually an additive variance over the other 2. Yeah, and that's it. So moving on to estimating and the issues, I would know those. Just know those, read through those 2 bullet points to know them. It's pretty basic stuff, but okay. moving on to genetics and ethics. So this was the one that you guys, I mean, it shocked me too. I was surprised that this was even going to be on the exam. But, you know, it's pretty basic and intuitive stuff, so it's actually kind of nice because if he does throw in a good amount of questions, this is like pretty easy compared to, you know, the other things. You've kind of talked about this stuff before, and it's pretty intuitive. So we're going to go into it. 23andMe, we had talked about that company, I think, for the first exam as an example, Dr. Hall had. So they got in trouble because they were advertising for, you know, personal genome sequencing. They send you a kit, you swab your mouth, send it back, and they'll send you your personal genome, like what you are predisposed to for diseases and things like that. But what they hadn't done was they hadn't gotten it approved by the FDA. The FDA hadn't tested it. They weren't advertising correctly to patients that, you know, these results are variable, that sometimes the tests are wrong. So people would get these tests back, and if they had a false negative result or a false positive, they would go and get these medications. They'd try to treat themselves. But if they didn't have the disease or they did have the disease, they didn't know. So there was a lot of confusion. I mean, there wasn't a lot, but it could have led to, you know, confusion among the people that were responding and buying these things. So the FDA kind of stormed in there and put a stop to it. They said that, you know, you can't market this, you can't sell it until we, you know, approve that this is efficient and that's accurate. And so just some guidelines that I think he wanted you to know, that the FDA requires that, you know, marketing for home tests for medical purposes, so it can be anything. It could be a pregnancy test, it could be a test for your blood pressure, whatever. Those things, the packaging needs to convey the risks and everything and what the results might mean. It also needs to be understandable, because a lot of times consumers aren't, you know, science majors like you guys. They're just regular people. But you know what I mean. So like if you have no background knowledge of science, or you have no access to knowledge of, you know, these things, and you don't know that, okay, you know, these tests, oh, they used PCR or whatever, like those get, you know, wonky results all the time, so who knows. So basic consumers need to understand what these tests are. Okay, objective versus subjective reasoning. So of course objective, you know, you have tangible evidence. Subjective, it's kind of, it's all relative basically. So there's no right answer, is that kind of a thing. All right, so ethics, it's just the definition of what it is right there under A. Ethical questions. So this is just kind of like a little bullet point overview of what kind of makes up ethical questions and how they're presented. And then these ethical terms down here, I would know these. I would know the differences, and they're pretty intuitive again, like, but it was kind of interesting to read through them because the example that he's using is basically murder. So, you know, I value life, one should not kill, thou shalt not kill. He could easily put, you know, maybe not those same ones, but something similar on the exam and ask you which ethical term that phrase is kind of highlighting. Is it morality, values, religion, ethics, logic, or not logic, legal? So I would just know those and kind of come up with some examples of your own. And of course, like I said, we will have a practice problem about that on the exam, the practice problems. Okay, and then justifying the ethical decisions. I would just read through this. I'm not going to go through it too much. You have an argument, which is a claim, justification, a warrant, and then it's just showing you what weak justifications are consisting of, strong justifications. Read through it, understand it, maybe know a few. Again, this is all stuff that's so intuitive that you could probably come up with something on the spot on the test and it'd be right. So yeah. All right, moving on to the 4 principles of bioethics. Know these. Definitely know these, and know what they are. So memorize them, come up with a mnemonic if you need to. But yeah, you need to know a definition for them as well as knowing them. Okay, moving on. So genetic testing, just know the 4 different techniques that you can use. And then it's just showing you the categories of the types of genetic tests. Again, you don't need to necessarily memorize them, but read through them, understand what they are, maybe be able to name a few. I would say that you should memorize the techniques, chromosomal, molecular, biochemical linkage. Gosh. But don't memorize the categories. Just maybe know a few in case you're asked about it. Genetic tests vary in their usefulness, and these are the 2 reasons why I would know that. It's a fair question. It's good to understand. Oh, and moreover than that, there's a chart that'll show you high clinical validity over low clinical validity, and how they like correlate to the to different categories of effectiveness. So obviously, if you get in quadrant 2, you're getting the best genetic test is what that is. And I think he has like for the next like slides that I put in here. So I'll go ahead and talk about those real quick. I would know those diseases at length. I don't think you need to know the penetrance and stuff like that, but go through and redo these examples on here. So like under it, he has a question, what category of genetic test would this be? Kind of go through and do those on your own and try and figure out, what category these fall under. And I would know them at length. I would know that hemochromatosis is a defect of the HFE gene because it's a mutation in a protein that regulates iron absorption from food. So I would know just that much. In case, like you see it again on the exam in this format and he asks you a question about it. If he told you guys that you don't need to know the diseases themselves, then don't worry about it. But I have not heard that. So I'm going to go ahead and say that that's the most you should know, which is not too much, but just in case. And then here, I didn't want to retype it because it's not terribly important, and it's not, but genetic counselor, what do they do, and what kind of training and things like that. Just read through that. I don't know. That's probably going to be a true-false question or something. I don't know. Okay. So, yeah, I think that's about it because I already went through those slides. So that's it. I kept it to, what does that say, 37? Okay. Well, still less than an hour. I was trying to do 30 minutes, but it's okay. Okay, so what we're going to do is now that you guys kind of have an idea of what's the more important stuff from the packet, we'll have a practice problem set for you at noon. Good luck on your test, guys. Thanks for everything this semester. You guys have been awesome. And yeah, best of luck to you and everything else that you do. Bye.